Hello everyone, we are group 10. We are going to talk about TLR4. Before discussing the TLR4 receptor, let's talk about innate immunity. In innate immunity, when dendritic cells recognize a pathogen, they release cytokines and interferon to cause inflammation. TLR4 is very important in this innate immunity. Toll-like receptors are used by dendritic cells to recognize pathogens, so TLR4 is also one of the toll-like receptors. To briefly explain the origin of the name toll-like receptor, the gene encoding this receptor and the toll gene of Drosophila has similar base sequences, so the name was given. The toll gene is required to determine the dorsoventral axis and is required from the developmental stage. TLR receptors are divided into 10 groups according to the type of ligand. In the case of TLR4, it recognizes lipopolysaccharide of gram-negative bacteria and some others. Gram-negative bacteria are bacteria that do not stain purple by gram stain, and there is an outer membrane made of lipopolysaccharide on the outside of the peptidoglycan layer. When the TLR receptor recognizes a ligand, signal transduction is transmitted into the cell, transcription in the nucleus is regulated, and the substance that acts is produced. Let's take a closer look at each step. First, the recognition of ligands. This section describes the case of LPS, or lipopolysaccharide. LBP, or LPS binding protein, binds to LPS of gram-negative bacteria to take out LPS and thereby to CD14. CD14 transports LPS to TLR4, which can recognize LPS with the help of MD2. At this point, once a complex of TLR4, MD2, and CD14 is formed, another set of TLR4 and MD2 will come closer to form a dimer. Next is transcriptional regulation. By forming a dimer in a round shape, tau IL-1 receptors in the cell also form a dimer. MYD88 recognizes and binds to this structural change, from which signal transduction goes downstream, and finally the transcription factor NF-kappa B is produced. NF-kappa B enters the nucleus and binds to target genes to promote transcription into mRNA. Finally, when the production of mRNA is promoted, substances such as cytokines are produced by translation and released to the outside of the cell, resulting in innate immunity. Finally, I would like to introduce state of illness related to TLR4. Overproduction of various inflammatory cytokines such as TNF-alpha, IL-6, and IL-8 by TLR4 signaling may cause endotoxic shock. Symptoms of endotoxic shock include fever, chills, leukopenia or decreased number of white blood cells in the blood, bleeding, hypotension, tachycardia, multiple organ failure, and stroke. That's all for our presentation. Thank you for listening.